Hello there, and welcome to another episode of Plant Stars, the talk show where I introduce you to my house plants. Today, we're going to meet some roots. No, not the band that Questlove is in, Plant Roots. So first I'm going to show you this little avocado pit that I've been growing. So this is just a pit that I took out of an avocado that I bought at the grocery store, and I stuck some sticks in it, and it's been hanging in some water, and you can see that roots have started to form out of the pit. If you touch these roots, they're kind of slimy. We're going to talk about today why the roots are kind of slimy. Here's the tip of the root, and hopefully, if I grow this long enough, it'll turn into a little avocado plant. Probably will never ever fruit, but here's one root in my house. Let's put that back in the cup. Ew, very slimy. Another root that I'd like to introduce you to is on this orchid. So in my bathroom, in my house, I have a skylight that provides a lot of light into the bathroom, and it's a perfect environment for growing orchids, which like really hot, steamy environments. And coming out here is an example of an aerial root, and we'll talk about what those function in too, and why they grow so well in my hot, humid bathroom. Another root I'd like to introduce you to is on this little succulent friend that I got from one of my roller derby friends. So this is just sitting in a cup of water right here. This is a clipping, and the roots that are growing out of this plant are called adventitious roots, and we're going to talk about those today too. But this is just a little clipping that didn't have any roots at all when I got it, and now because it's been sitting in a cup of water, it's grown this really nice root system out of it called adventitious roots. And it's even got a cute little flower on top that we're going to talk about flowers today too. So, let's get to it. Let's talk about plants. That's not a plant. That's a nudibranch. Okay, so let's start by finishing off our discussion of phloem transport and talking about phloem and xylem, and then we'll get to talking about the roots. So sugars are produced in, and we're going to talk about sink and source in a little bit, but sugars in plants are produced in sources, which are things like leaves, and then those sugars need to be delivered to growing parts of the plant via the phloem in a process that we call translocation. And translocation is basically the movement of sugar through the plant. That sugary solution that's moved through the phloem is called the sap, and that's what a lot of plant-feeding insects feed on, like the mealybugs that are living in my house. This sap that's in the phloem is going to provide the suitable carbohydrate building blocks for the roots and other actively growing regions of the plant. The most widely accepted model for carbohydrate transport through the phloem is called the pressure flow hypothesis. This is where dissolved carbohydrates flow from a source to a sink, where they're then unloaded and used by that plant tissue. So the carb sources include tissues like mesophyll cells of leaves, because this is where photosynthesis is occurring in the plant. Food storage tissues are going to be the sinks. Those are going to be growing tips of roots and stems and fruit. And then there's phloem loading. Phloem loading is when carbs enter the sieve tubes in the smallest veins at that source that we talked about. And then some of that sucrose travels from mesophyll cells to the companion and sieve cells via the symplast. And then most of the sugars arrive at the sieve cell through apoplastic transport. And this energy requiring step is driven by a proton pump. And so that's active transport of the sugars into the phloem. And these sieve cells have to be alive to participate in active transport because it's a proton pump that's involved in transporting these sugars. But bulk flow is going to occur in the sieve cells without any additional energy requirements, and that's because of the difference in water potential in the sieve tubes and that of the nearby xylem cells. So water flows into the sieve tubes via osmosis. So it's passive movement of water from the xylem and then active transport of sugar into the phloem. There we go. Okay. So let's talk about this source and sink that I mentioned before. This diagram that you're seeing here is a diagram of mass flow through the plant. The red dots represent sucrose molecules, and then the blue dots are water molecules. After moving from the mesophyll cells of a leaf or another part of the plant into the conducting cells of the phloem, the sugar molecules are transported to other parts of the plant by mass flow and unloaded where they're required. So at the sink, the sucrose and hormones are actively removed from sieve tubes, and then water follows by osmosis. And then the turgor pressure at the sink is going to drop, and that causing a mass flow from the stronger pressure at the source to the weaker pressure at the sink. And most of the water at the sink then diffuses back into the xylem, where it may be either recirculated or lost through transpiration. So just in summary, this is just a nice slide to summarize what's going on in xylem versus the phloem. You've got the flow of carbohydrates going through the phloem. You've got the flow of water happening through the xylem. Water and carbohydrates are traveling to all parts of the plant, but again, xylem is only flowing up. Carbohydrates are moving through the phloem in that sugary rich solution called sap. Waters and minerals are going to pass through the xylem along a gradient of increasing negative water potential, which we talked about in the last lecture video. Then water exits through the plant through the stomata and the leaves, and that's where you create that negative, the water potential of the air, which is negative, and then water enters the plant through the roots before it exits out the stoma, and then the water potential of the soil is close to zero, and all this is creating that movement. That water potential is creating the movement of the water through the plant. And again, in phloem, you've got active transport of sucrose happening in phloem through proton pumps, and then passive transport in the xylem of the water through osmosis. Now let's get to the roots. Let's talk a little bit about the basic external structure of the roots. The roots, their main function is to anchor the plants to the ground, and they're involved in the absorption of both minerals and water. These minerals can be really, really important for plant nutrition. And the basic structure is you have this primary root in the middle, then you have these lateral roots that are branching off. You have the root apex, where you've got a lot of growth and cell division occurring. And then, like I showed you on that little succulent plant that I have, you can also have adventitious root. These are plant roots that form from any non-root tissue. So like a cutting of a plant that then is able to grow roots from it, that's adventitious roots. And these are produced both during normal development and in response to stress conditions, so such as flooding or nutrient deprivation or wounding, as basically when you're taking a cutting of a plant and then trying to grow from that cutting, you're basically wounding the plant. And so those are adventitious roots. So let's talk a little bit about what's going on in the root tip where there's all this cell division happening. There are 4 regions that are commonly recognized in developing roots. That first region is called the root cap. The second region is called the zone of cell division. The third region is the zone of elongation. And then the fourth region is the zone of maturation. In the last 3 zones, the boundary isn't really well defined between these. But let's first talk about the root cap. So in the root cap, there's no equivalent of this piece of tissue that's happening in the stems. And this root cap is composed of 2 different types of cells. You have the intercolumnal cells that line the inside, and then you can have the lateral root cap cells. These root cap cells are continuously being replenished by the root apical meristem that you see here. And the main function of the root cap is to protect the delicate tissues that are growing behind it. Soil is maybe it may feel soft in your hands depending on the kind, but soil can be pretty abrasive. It's made of sand and other really harsh particles, and so this root cap is protecting those as it grows through the soil. The root cap also makes a substance called mucigel. So I told you when I was touching those avocado roots that are growing out of that seed that they felt slimy. That mucigel actually has a really important function in lubricating the roots and helping it glide through the soil so that it doesn't get damaged. And it also serves as a growth medium for any nitrogen-fixing bacteria. They're going to grow in that slimy gel that grows on it. The root cap also functions in the perception of gravity. So like if you tipped a plant on its side, the roots would still grow downwards towards the ground because of this perception of gravity that's happening here in the root cap. Next, we have this zone of cell division. Most of the activity here is taking place in the edges of the meristem. Here, where the cells are dividing about every 12 to 37 hours. You can also see division of the apical meristem daughter cells into two different tissue types here. Then we've got the zone of elongation. Here's where the roots lengthen because the cells are produced by the primary meristems are elongating here. That's why it's called the zone of elongation. So any cell division and cells that are created here then are elongated here. There's no further increase in cell size above this zone of elongation. And then you have the zone of maturation. This is where the cells are going to differentiate into specific cell types after elongation. So let's talk about how water gets into those roots and makes its way into the rest of the plant. The cells of the root surface cylinder mature into epidermal cells, and this includes both root hair and non-hair root cells. So let's talk about the different pathways of mineral transport that happen in this root. So the minerals are absorbed at the surface of the root, and in passing through the cortex, they either must follow the cell walls and the spaces between them or go directly through the plasma membranes and the protoplast of the cells, and they pass from one cell to the next by way of the plasmodesmata. When the minerals hit the endodermis, though, they can't get through the cell walls because they're blocked by this thing called the Casparian strips that you see in pink here. So what the Casparian strip is it's a waterproofing band that forces waters and minerals to pass through the plasma membranes of the endodermal cells rather than through the spaces. And that's the function of that Casparian strip. So it's not, those minerals aren't just getting lost, they're actually getting taken up by the cells. Both monocots and eucots have this Casparian strip so that nutrients are going directly into cells and not just flowing straight between the gaps in them in the cell where the cell wall is. Now let's talk a little bit about ion transport. So this is the movement of water into the root and ions. Let's talk a little bit more about those ions and those minerals that are important for plant nutrition. Active transport is going to be required to move positively charged ions into a root hair. And only minerals that are dissolved in water between all those soil particles are available for uptake by the roots. So those minerals have to be in water for the plant to be able to take them up. Both minerals and organic soil particles tend to have negative charges, so they attract positively charged molecules and ions, and then the negatively charged anions stay in solution, which creates a charge gradient between the soil solution and the root cells, so that positive ions would normally tend to move out of the cells. But the proton pumps move hydrogen ions out of the root to form a strong membrane potential. And that strong electrochemical gradient then causes potassium ions and other ions to enter via ion channels in the root. And some of those ions, especially the anions, are going to be used as co-transporters. And the membrane potential maintained by the root, as well as the water potential difference inside and outside the root, affects the root transport of those minerals. So this is how this plant is going to uptake the minerals through the roots from the soil. Now you can see a lot of specialization in root structures, and some of these are really, really cool. The primary function of the roots is to anchor the plants to the ground, so they can be the site of water and mineral absorption, but there are lots of different strong evolutionary pressures in the environment have created secondary functions for some of these roots, for some of these root support structures. So you can have support roots. For some plant, there are these special adventitious roots which grow directly out of the stem. So you can see this is not coming out of root tissue. This is coming directly out of a stem, so it's adventitious. They act to support or hold up the plant. This right here is a prop root on a corn stalk. Corn's really, really tall and very thin for a plant, and so these prop roots help it stand up. Some trees, this here is a tropical fig tree. Some trees have large buttress roots, which help to keep the plant upright. You see a lot of buttress tree roots in tropical plants because tropical regions are really hot, they're really humid, and because of that, and we'll talk about this later when we get to ecology, because of that, nutrients are constantly cycling in the environment. Any leaves that fall and decay on the ground are very quickly being broken up and then reused by plants that are growing in it, which means that soil is really thin because it's decomposing and being cycled really, really fast. And if the soil's really thin, it's not providing a lot of structure for the plants to stand upright. And so what a lot of tropical plants have done to deal with this thin and loose soil that's in the tropics is they create these buttress structures that help them stand up in this really thin soil layer. So you see a lot of really cool structures like this in trees in the tropics. A lot of figs do this. One of my favorite trees that has buttresses is called a strangler fig that wraps itself around other trees. You should Google it, they're cool. You can also have aerial roots. So when I showed you in my little talk show at the beginning, my little orchid plant that grows in my bathroom, they have these aerial roots. So some plants, orchids specifically, they usually live high off the ground in tropical regions, and they'll live on tree branches or on rocks. And then those types of plants that do that need roots that can attach to the ground, but they won't have roots that attach to the ground, but they'll have roots that can collect moisture directly from the air. So in my bathroom, what I talked about that has that skylight, every time I take a shower in there and I make it really hot and humid, the aerial roots of my orchids that are growing in there will then collect moisture directly from the air. So that's why all my orchids are growing really well in there. You can also have these really cool things called nematophores. For plants that live in very wet or water-saturated environments, the roots, they do actually need access to air. If they become waterlogged, they can rot. So a lot of them will grow specialized root structures which extend above the water level to allow for gas exchange. This picture here, these are, they're called knees. These are bald cypress tree knees, and these are the root structures that are extending up out of the water to allow for gas exchange. So some really cool evolution that's happening in plants that I love talking about. Some roots are specialized for storage. So roots may be the location of carb storage for plants. So one example is red beets or sugar beets or carrots. We'll talk about tubers in a second. These are not tubers. You can tell the difference between a tuber and a storage root because of the presence of growth rings. So you can see here on this cut piece of carrot, there's growth rings. And that gives you an indication that these are root tissues and not tubers. So in a potato, you don't see growth rings, so they're not actual root structures. Water storage may also occur in roots. Many of the plants that live in arid or dry climates, the storage of water in the roots is going to allow that plant to survive even when there's little or no water available. One example of this is cucumbers and melons. They're able to grow in dry, sandy soil because they store a lot of water in their roots. In this picture, you can see the huge root structures here that are storing water for plants. Another modification of roots, so this is more of a this is a symbiotic relationship that has evolved in some groups of plants with a symbiotic bacteria. So in a couple of weeks, we're going to talk about mutualism, but a mutualism is a type of symbiotic relationship which benefits both of the species that interact with each other. And in this example of rhizobium, the bacteria which live in the nodules of legume roots benefit both organisms. So the nitrogen fixation of the bacteria assists the plant in uptaking nutrients from the soil, and then the carbohydrates that the plant produces then feed that rhizobium bacteria. So that gel that the mucigel that we talked about earlier, that kind of slimy stuff that's on the base of the plants, that can serve as a carbohydrate-rich source for bacteria to then eat and grow as they're fixing nitrogen from the soil that then makes those nutrients available for the plant. So if any of you pulled up your pea plants that you were growing, you might have seen these little nodules that were growing on the roots of your pea plants from GenBio Lab. Those were little nodules of bacteria growing in them. There's also some symbiotic fungi that will grow and infect root systems. These are fungi infect about like 90% of the root systems of vascular plants. Not the same limitation is found with the legumes and the rhizobium. This fungus acts to increase the surface area of the roots and assist the plant in the uptake of water and minerals in the soil. And then the plant, in turn, like it does for the bacterium, provides food for the fungus, usually in the form of carbohydrates. So a lot of these fungus or bacterial mutualisms that you'll see on plant roots, what the plant is providing in the symbiosis is carbohydrate source. And then the fungus or the bacteria is allowing for better absorption of water and minerals from the environment. So rhizobium, symbiotic bacteria. In this example, the bacteria are actually fixing nitrogen. With the mycorrhiza, which are fungus, they are increasing the surface area of the roots to increase the uptake of water and minerals. So they're not actually doing any sort of fixing of the nitrogen here. Now let's talk a little bit about shoots and stems and talk about the external structures. As we already talked about, the apex of the shoot will be the primary site of growth in the stem. This is the location of the meristem stem cells. This growth is going to provide a terminal bud, which is the location of the extension of the stem. And as the stem grows at regular intervals, the meristem cells will form bulges on the sides of the stem. And then these bulges are the areas where the leaves, flowers, and branches will form along the stem. The location of these bulges that you'll see will provide the attachment for the leaves, and this will be the location of the nodes that are labeled here, with the empty stalk between the nodes known as the internode. In addition to the location of the leaf attachment, there will also be auxiliary buds located at the nodes. These will serve to replace the leaves if they get damaged, or they'll form a site for branching if the other branches become damaged. For woody plants such as deciduous trees, and by deciduous I mean trees that will shed their leaves in the winter, the loss of the leaf will leave a scar on the surface of the stem, and as a result of the regulation of hormones in the plant, this scar tissue is not dead tissue, but may be reactivated in the spring to allow for growth of the new leaf. Now let's talk a little bit about the internal structures of these. We've already mentioned the difference between the eucots and the monocots with respect to the organization of the and location of the vascular tissues. But one thing else to point out is the location of the ground tissues and the eudicots. There is ground tissue found on both sides of the vascular rings and the stems. The inner ground tissue forms the pith, and the outer will form the cortex. Yeah. Oh, and then the cortex. So the point of bringing that up is that the cortex is going to be the location of the meristems where they're going to provide secondary growth. So secondary growth is a phenomenon that only happens in eudicots. And because these eudicots have this ability for second growth, they're generally tend to be larger and more complex in structure than monocots. Before secondary growth can begin in eudicot stems, primary tissues continue to elongate as the apical meristems produce primary growth, and as secondary growth begins, the vascular cambium is going to produce secondary tissues, and that's when the stem's diameter is going to increase. And then in this diagram here in C, this is a 4-year-old stem of a plant, and you can see here that as the secondary tissues continue to widen, the trunk has become thick and woody. And again, this secondary growth you're only going to see in eudicots. So those are basic external and internal structures of the stem. Now let's talk about some really cool evolutionary modifications of the stem that have evolved in different groups of plants. One modification you can have is a bulb. This is just a below ground. Some plants will have a bundle of fleshy leaves which will be attached to a small knob-like stem. These are common in onions and lilies and will be important in the regrowth for next year. And the leaves of the bulb are going to protect the meristem cells, which will differentiate to form the leaves above ground in the next generation. You can also have rhizomes. These are horizontal stems which grow underground, and they'll contain small scale-like leaves underground, and these will contain a node and auxiliary bud. These generally grow near the surface of the soil and are able to produce photosynthetic leaves at regular intervals, which will grow above ground. And these are common means of growth for grasses and ferns. You can also have stolon or runners. These are modifications of the stem which can grow a pretty far distance away from the main plant, and where the node is placed, and they're able to grow adventitious roots from these structures, which will then anchor the plant and allow it to propagate. So these are genetically identical asexual buddings. These stolons are asexual buddings off the main plant. The stolon are formed in a location where there are long stems with only a few nodes present. Unlike the runners, these are not able to form roots. And then you can have tubers, which are down here. Tubers are modifications of the stolon, in which the location of the stolon on the stem will become swollen with carbohydrate storage. These are going to contain a number of auxiliary buds, which will be able to grow if the rest of the plant dies. So one example is a potato, and the eyes of the potato are the auxiliary buds that are formed along the stem modification. You can also have tendrils, so some plants like grapes and my passion vine, my passion flower vine that I showed you in the last video, they'll form from modifications of the leaves, which are able to grow out and attach the plant to supporting structures. And as you may expect from things like tendrils, they'll function to allow the plant support by attaching to other plants or to artificial structures. So my passion flower vine that's growing in my house, I have a pole made of bamboo that's wrapped in moss that is the moss kind of creates like a rough surface for those tendrils to attach to help support the plants. I also actually have a pepper plant that black peppers come from. That's also a viney thing that sends out tendrils. And then you can have these cladophylls. This is a modification of a stems where you get a flattening of the stem. These are really commonly found in cacti. These flattened stems will be the photosynthetic and water storage areas of the plants. So these are the pads that you see in a cactus. Part of those are leaves, and then they form the cladophyle. So you'll notice that the spines on the cactus leaf are on the cacti are leaf modifications. So let me go over that again because I think I said it a little confusingly. Cladophylls are modifications of stems that are flattened, and these are found in cacti. These flattened stems will be where they do photosynthesis and water storage in the plants. So now let's talk about leaves. We've talked about the roots, we've talked about the stems, we're working our way up the plant to the leaves. For most of the plants, leaves are going to be the primary photosynthetic organ or tissue of the plant. And since these are extensions of the stem, they'll generally have a flat surface that's covered in photosynthetic cells, which are going to provide energy for the plants. There are two main classes of leaves. You can have microphyll leaves, which these leaves only contain one single vein and they don't extend to the end of the leaf. The vein does not extend to the end of the leaf. These leaves are generally small and are not a common leaf type. And then you can have megaphyll. This is what most plants have. These are leaves that contain multiple veins, like you can see here. So this is a microphyll. This is, This is a megaphyll. There's no microphyll on here. Okay. So what are the veins that are in the plant? Veins contain the vascular bundles of the leaves, and they're also important for the support and connections of the leaflets. They're connected to the stem of the plant by the petiole at the base here. And there can be differential arrangement of the veins. You can have monocots, which have parallel veins. That's what's going on down here. Monocots lack petioles. This is a monocot leaf. It's got those parallel veins running here. And then in eudicots, you can have net-like veins. Very, very obvious here. Eudicots, they're going to contain a network of different branches of veins throughout that. So with the monocot, things like grasses are really classic examples because of those parallel veins that you can see in the plant. Now in eudicots, you can have simple versus compound leaves. This is really important to know for when you eventually take my ecology class because it's really important for identifying different species of trees. Locally here in Pennsylvania is knowing the difference between a simple and a compound leaf. And you can tell the difference between these by the way the leaflets are connected to each other. That's a leaflet there. If all the leaflets form connected blades between the veins, this would be a simple leaf. So you can think of the leaf of an oak tree when you think of this. If the leaflets are not connected directly, but rather there are gaps between the leaflets, then this would be a compound leaf. Examples of these would be elders, walnut, and ash trees, box elder trees. Now let's talk a little bit about the internal structure. That was the external structure of leaves. Now let's talk a little bit about the internal structure. The top and bottom of the leaves are very different, and you can see this difference within the leaf as well. The arrangements of the sides, the tops and the bottoms of the leaves will be determined by the genes that are going to be expressed, and therefore the proteins that then are made in the different parts of the leaves during development. So let's talk about the layer within the leaf, starting from the top down. The outer layer of the top of the leaf will be the cuticle. This will be dermal tissue outside the leaf, and it's waxy, as we mentioned before, to prevent water loss. The epidermal cells under the cuticle are almost clear, which prevents the loss of light and energy as light passes through to the ground tissue. Under the dermal layer, there are generally two layers of mesophyll cells. These middle layers will be made up of the ground tissues. And then you have the palisade mesophyll. These are columnar cells which are going to give support to the cells, and they're going to contain a lot of the parenchymal cells with the chloroplasts. These types of cells are also referred to as chlorenchymal cells, which are a special class of parenchymal cells. So that's the palisade mesophyll. You can also have the spongy mesophyll. This is a less densely packed area of the leaf below the palisade mesophyll, and this area will be more open to allow for gas exchange within the leaf. And then below this spongy mesophyll layer, there's a lower layer of dermal tissue. This is going to be similar to the top layer, with one exception. This is going to be the location of the guard cells and the stoma the stoma of the leaves, which will be the site of gas exchange. So let's talk a little bit about the difference between C3 and C4 leaf structure, which we've covered a little bit before, but let's go over it again. C4 plants are going to have to overcome the limitation of photorespiration. Well, how they overcome it is by the physical separation between the location of carbon fixation and the Calvin cycle. We talked about that, how they separate out where they do these two things. And then you can see this organization in the actual photosynthetic cells inside the leaves. So you'll notice for the C3 plant, there's no separation between the two, we don't have separation between the two processes. The cells that contain the photosynthetic activity are located throughout both layers of the mesophyll, and so there's no separation between the function of the cells. You can kind of see them. The mesophyll and the bundle sheath cells are all mixed up in here. But then in C4 leaves, the location of the chlorophyll containing cells is going to be further away from the surface of the cell, and that's because these are the cells which contain the Calvin cycle, and they're further away from the location of gas exchange and closer to the location of water near the vascular tissue. Because these cells will form a sheath or a cover around the vascular bundle, they're referred to as the bundle sheath cells. And then you should notice that in the C3 plant, not only do they lack that particular location of this photosynthesis bundle sheath cells, they don't actually contain any vascular bundles. So now let's talk about some leaf modifications. One example are floral leaves. In some plants like poinsettias that you'll see around Christmas, they don't actually produce flower petals. Rather, they just change the pigments that are in their leaves at the top of the plant to serve the same function. So the whole point of flower petals is to attract pollinators to the plant to collect pollen and then transfer it to another plant so that they can reproduce. And so poinsettias just take their leaves, change them a color, and it serves the same role as flower petals. You can also have spines like you have in cactus. For many plants that live in dry areas, they need to protect against water loss. And so the leaves will develop into spines rather than flat leaves. So you should note that these are plants with the cladophylls that we talked about earlier, those flattened photosynthetic stems, which will take the function out of the leaves. So the flat pads of a cactus, like I was saying before, are the cladophylls. Those are the stems. And then the spines are the actual leaves that don't do any photosynthesis anymore. And these help protect against water loss. You can also have shade leaves. For many trees, the amount of light which passes through the upper layers of the leaves will be limited, and therefore the leaves have to develop to increase the light which they are able to collect. And so the leaves can actually change the orientation relative to the sun so that they can increase their exposure to the light or decrease it depending on if they're getting too much or too little light exposure. Also, you can have lower leaves. So if a plant is tall in height, you can also have lower leaves that are developed to increase the light that they're able to collect. So shade leaves typically will have an increased surface area so that they can collect more light on lower areas of the plant. And you can see that here in these shade leaves here. You can also have insectivore leaves. Some plants don't rely on the sun alone for this source of energy and the soil. So what we've talked about so far is plants get their energy and their nutrients from the sun and then from water in the soil. Some plants in areas with really nutrient poor soil, and maybe they're not also getting a lot of light, have developed to eat insects as their nutrition source, which is really, really cool. And when you take my ecology class, we'll go out to Spruce Flats Bog and we will go see some of these really cool plants that live in the bog, because the bog is waterlogged and has really poor nutrition. So what they do is that they have these kind of lures, either by looking like a rotten piece of meat, by being purple and sometimes they stink, or by having these guides that draw the insects in, and then they either close up around them or they fall in, like in these pitcher plants, and they fall into a liquid and they drown and die. And then the plant produces enzymes that which then break down the insect and digest it. I think carnivorous plants are really, really cool. I love them. And that's it. That's the end of the structure and function lectures. And so tomorrow, Friday, I will post up the plant response and hormones section. Okay, talk to you soon. Bye.